Hi, I'm Karen Vanderwolf, a 2020 recipient of the Weston Family Boreal Research Fellowship with Wildlife Conservation Society Canada. I received the fellowship when I was a PhD candidate at Trent University. So my project is looking at the skin chemistry of bats in relation to the bacteria and fungi that live on their skin, and all of this in the context of disease susceptibility. So little brown bats were formerly one of the most common bat species in North America, until 2006, when a fungus was introduced from Eurasia to North America, which causes a devastating disease called white nose syndrome. It affects bats during the winter when they're hibernating. It doesn't affect all individuals or bat species equally though, and we don't really know why, which is why I initiated this research project. So I started my research on bats and white nose syndrome in eastern North America, which is where the disease first established. It's now found as far west as Alberta in Canada, but is not yet found in the Yukon. That's why I wanted to do a project there to get baseline data on the skin health of bats before the disease arrives. Yukon still has healthy bat populations, many of which are found in bat boxes. I was lucky enough this past summer to work with local government scientists to look at bat colonies across southern Yukon. Many of the colonies are in bat boxes, and some of them contain hundreds of bats that produce a lot of guano. After working in eastern North America for so many years, it was really great to see these healthy bat populations in the Yukon. To catch the bats, we would hang a harp trap at the bottom of the bat boxes where the bats exit at twilight. Harp traps are made up of a frame with fishing lines strung between the frames very tightly so that when the bats hit the fishing line, they fall into the bag below unharmed and we can collect them. When bat colonies are in buildings, we use mist nets, which is something that bat or bird biologists also use. The mist nets are very fine nests, kind of like mist, that are strung between two poles, which we then put in areas where we think the bats are going to fly. Sometimes the bats don't always emerge from their roosts when we think they're going to emerge, but then it gives us opportunities to catch up on our reading. When we catch the bats, we measure their size and weight, identify the species and sex, and then I measure the skin chemistry using probes. So specifically, I'm measuring the pH. We also swab the wings to detect the bacteria and fungi on the skin of bats. The mosquitoes were always very interested in our research and would keep watch with us throughout the night. Our preliminary results indicate that the bat skin chemistry varies depending on the roost type. And by that I mean roosts like bat boxes versus buildings. But overall, the skin chemistry of bats in the Yukon is quite similar to bats in eastern North America. So our lab work looking at the bacteria and fungi on the skin of bats is still ongoing, so we don't have any preliminary results from those samples quite yet. So our project provides baseline data on bat skin health ahead of the expected arrival of white nose syndrome in the Yukon. The project would not have been possible without these great collaborations with local scientists in the Yukon and, of course, with the support of the Weston Family Foundation. Thank you.